the end of muscle contraction, or what makes muscle contraction happen, is the calcium being released. So we need to figure out how we got calcium released from the sarcoplasmic reticulum. We are debating whether it's mechanically gated or whether it's voltage gated. And actually, both kinds of channels are involved in the process of getting us to muscle contraction. But actually releasing calcium at the very end of the process, the kind of channel that ultimately helps me to release the calcium from the sarcoplasmic reticulum is actually these mechanically gated channels. So we're going to work our way backwards from here. So mechanically gated calcium channels open up, and that's what's going to release the calcium. The tricky thing about the mechanically gated channels is how they open up. So this word mechanically gated here, I'm so sorry. Evelyn,
no, that's not a
maybe I need you to run downstairs for me, please. Can you go downstairs, please? Okay. Mechanically gated, remind me, when I talk about things that are mechanically gated in general, what does mechanically gated mean? How do I open those ones?
Okay, so something like physically pushes it, right?
Exactly. Yes. Yeah. So mechanically gated channels are ones that are pushed open. So we're looking for something that our step above this pushes the gate open. So the way that we are pushing the gate open is actually using a different kind of channel. So the kind of channel that's actually pushing this gate open is a voltage gated channel.
Oh, okay.
Voltage gated channel. Now, the voltage gated channel technically is not a channel for calcium, it's actually a channel for sodium. So a voltage gated sodium channel is opening up, and when this one opens up, it's actually attached to the mechanically gated calcium channel, which the mechanically gated calcium channel is the one that opens up the sarcoplasmic reticulum. Now, this voltage-gated sodium channel that's opening, this is one, here I'll help you out, Elaine. Those structures that we were looking for, the name of the places where the membrane folds and makes tubes, they're called the T-tubules. So the T-tubules are covered in these voltage-gated sodium channels. The function of voltage-gated sodium channels in general, so we'll put that here, voltage-gated sodium channels. Generally, their job is to change the charge on the plasma membrane to make the thing called an action potential. So these channels make action potentials. So if you remember from your studying and your reading, an action potential is where I get the message to contract, and then I have to send that message all over the membrane. So voltage-gated sodium channels are what I use from the point that I get the message to send that message everywhere. I'm going to use voltage-gated sodium channels to do that. So what opens up the voltage-gated sodium channel that's attached to the mechanically gated calcium channel is when that change in membrane charge comes. So these guys opened up. We'll put a note up here. We had an action potential. We had a change in membrane charge, so membrane charge change. And that leads to these channels opening up. And when they open up, they push on the mechanically gated calcium channels to open them up. We'll take one more step back too, though. The process of muscle contraction, we've talked about how it uses voltage-gated channels, how it uses mechanically gated channels. It also actually uses chemically gated channels. The chemically gated channels are found at the place called the neuromuscular junction, where a neuron and a muscle cell meet. Do you happen to remember the name of the chemical that I used to open up those chemically gated channels?
Darn, is it acetylcholine?
Yes, yeah. So our abbreviation was ACH, right? ACH, acetylcholine.
Okay, is it just acetylcholine or the one with the E at the end of it, or?
So that one's different. Yeah, we can talk about that one too. That is different from it. Acetylcholine is the name of the chemical that opens up these channels. Once I open up the channels at the neuromuscular junction, I'm going to use that to build an action potential, which is going to open up voltage-gated channels to move that message. Some of those voltage-gated channels are attached to mechanically gated channels, and that's going to lead me to release my calcium. So the question that you started with, where we actually needed to get to right from the homework, was that it is mechanically gated calcium channels. But the process of muscle contraction does use all three types, just at different places.
Okay. Thank you. That's helpful.
Okay. And you mentioned ACH with the E on the end, right? So let's talk about that. That's a little bit different. So ACHE is an abbreviation for acetyl, let's see if I can spell it, acetylcholine esterase. Acetylcholine esterase. This ends with, we have an ACE here at the end. So the ACE tells us that it's an enzyme. Do you happen to remember what the enzyme acetylcholine esterase, what its job was, what it does?
Does it
Or we've got a couple of
break down something? Does it break down?
Yeah, it does. Yep, it breaks something down.
Can it break the calcium down? Or?
Actually, its name tells me what it breaks down.
It breaks down acetylcholine.
Yeah, exactly. Yes. Yeah. It seems like a much harder question than it actually is, right? So this is an enzyme that breaks down acetylcholine. So once so the process of muscle contraction, we open up the chemically gated channels using acetylcholine. When I open up those channels, that helps me to get my membrane charge changed. And I'll send that everywhere throughout the muscle cell using these voltage-gated channels, which is going to lead to my calcium releasing and then the whole muscle contraction process. The thing is, once I have sent this acetylcholine, once I've received that message and I've started to tell my muscle cell to contract, I don't want to keep sending that message forever. So I only want my muscle to contract once from each acetylcholine chemical that's released. So once I contract it, then acetylcholine esterase is going to break it down and make sure that I don't keep contracting over and over and over again because of the same acetylcholine chemical. I only want to contract once when I release it once. This is the enzyme that breaks it down to make sure I only contract once from that chemical.
Okay. So is that kind of the one that when like sarin gas gets when someone's exposed to sarin gas, like acetylcholine esterase can't break it down, right?
Exactly. Yep. And so if acetylcholine esterase can't break it down, then I sent an acetylcholine message 5 minutes ago, and I'm still responding to the same acetylcholine message that I sent 5 minutes ago. So that's why when you're reading about some of those signs of someone who's been exposed to it, things like muscle spasms or uncontrolled muscle contraction, that's why. Because I should have broken down this message 5 minutes ago, but I didn't because acetylcholine esterase isn't working anymore. So it's absolutely right. Yep, so I'll put a note here on our slide that sarin deactivates acetylcholine esterase. That's a long word.
All right, well, thank you.
Yeah, you're welcome. Maria, I saw you pop in too. Do you have any particular questions I can help you out with?
No, I just show up to see what others have for questions, and this was really helpful too. This actually was one of the questions.
Yes, yeah, and what I'll say too is it's really important to make sure that we understand what happens with sarin gas and what happens with Botox, because both of those will be on the exam. So we just talked about sarin gas, right? How sarin gas is gonna stop my enzyme from breaking stuff down. And if I can't break it down, then I keep responding over and over again to acetylcholine. Have we read about Botox yet? Has anyone read about Botox yet?
Yes, I did.
I'm trying to remember what it blocks though. It blocks it breaks down the I can't remember what they're called. It like cuts them up, it like chops something up, and then it
Yep, yeah, so
blocks
it
another
chops
thing
up some
for.
proteins for me. So here first I'll put its name for us, botulinum toxin. That's what Botox stands for. So botulinum toxin, it chops up proteins, and we'll be specific here, proteins that help with the release of acetylcholine. So inside the cell, we store acetylcholine in these little circles. The picture showed us the little circles here. These little circles are called vesicles. They're kind of like bubbles. And so I make acetylcholine and I store it inside these little vesicles, these little bubbles. If I'm going to release that acetylcholine, I need to have these special proteins that are there on the membrane. So what Botox does is it goes in and it chops up those proteins. And now I've got these bubbles that are full of acetylcholine that I can't do anything with because I can't attach them to the membrane and I can't release them. So Botox prevents me from releasing any acetylcholine. When we talk about sarin gas, that prevents me from ever breaking down acetylcholine. So they're kind of the opposite problem of each other.
Okay.
What else did we want to chat through? It doesn't, we don't even have to have specific questions. I'm totally willing to just talk about stuff with you guys if we have some ideas we want to talk about.
Let's see.
I'll pull up my lesson.
I guess one of the things I'm trying to work on is like how you walked through the action potential of of muscle contraction. Like I just need to work on like understanding it more and like being able to like explain it. But I'll have to study that.
Yes. Here, let me see if I can pull up. I know we have some good pictures from this lesson. Here, I'll pull up the one. It's kind of a summary picture. This is a really good one when we talk about being able to explain things in your own words, right? This is a really good one for us to try to be able to explain in our own words. So this one shows me everything, the whole process of muscle contraction, from the beginning where I get a message to contract my muscle, all the way down to the end where we do the muscle contraction. So let's make a few notes for ourselves here on our picture to help us. I don't know, what page is this on your current outline? I don't have my page numbers in front of me.
Page 10.
Page 10. Okay. So on page 10, I think I even told you to explain it in your own words, so we can explain it together. Let's start here at the beginning with the process of muscle contraction. Here at the beginning of muscle contraction, here's those things. Let me circle it that we were just talking about right there. We call them vesicles, right? Bubbles. So I'm looking at right here a motor neuron, which is a neuron that can send messages to a muscle cell. So inside that motor neuron, it has all these little bubbles that are holding the messages. They're holding acetylcholine to tell the muscle to contract. So when my neuron decides that it's time for the muscle to contract, what it's going to do is take this bubble of neurotransmitters, and it's going to fuse that bubble with the plasma membrane of itself, spit out those. So you can see all the little green dots down here. All of these are acetylcholine chemicals that have been released by this neuron. So those little green spots that go down attach to, and it's kind of hard to see. I apologize. My resolution's bad. Let's see if we can zoom in a little bit. Okay. They attach to these little green channels right here. And these are the first kind of channels that are involved in the process of muscle contraction. These ones open up with acetylcholine. So acetylcholine is a type of chemical, which means that these ones that I see in my picture right here, these purple tubes, which kind of gated channels are those ones? What opens them?
Chemical gated
Yeah,
channel.
they're chemically gated, right? Chemically gated.
Oh,
Chemically gated.
gated.
Yep, because we're opening it with those chemicals with acetylcholine.
That's right.
Chemically gated, and these are going to be sodium, chemically gated sodium channels.
Okay.
So these are the ones that are found at this place. I guess it's not labeled. Let's add this label to it. This image shows me the neuromuscular junction, the place where a neuron meets a muscle cell. That's what I see right here. So the neuron released these little chemicals, these little spots that I see here. These spots, these chemicals went down and opened up chemically gated sodium channels. When chemically gated sodium channels open up, let's see if we can remember this. Does sodium normally, before these channels open up, is there usually more sodium inside a cell or outside a cell? Where is there usually more sodium?
Outside.
Yeah, sodium is outside, right? Sodium is something, here we'll draw ourselves a little cell. Here's my cell. Sodium is one of the things I have a whole lot of outside the cell. I also have a whole lot of chloride outside the cell. Oops. The ion that I have inside, do you remember which one we have a bunch of inside?
Potassium.
Yeah, that was the potassium. Okay, K plus in there. Potassium inside. So outside my cell, I've got a bunch of sodium and chloride. Inside my cell, I've got a bunch of potassium. The reason that that matters is because when I open up these chemically gated sodium channels, my sodium wants to rush inside. So sodium that I've got a bunch of outside my muscle cell now is going to rush inside my muscle cell. And when that sodium rushes inside the muscle cell, just like we saw in unit number 1, it's going to do something that we call depolarize the membrane. So the membrane charge is going to goes up. Membrane charge goes up. So what happens is this place on my membrane that used to be very negative. It used to be negative 90 millivolts, so it's very negative in here. I brought in a whole bunch of these positive sodiums because I opened up a chemically gated channel for them. And when all of those positive sodiums came in, my membrane charge went up. So at the site of the neuromuscular junction, we have chemically gated sodium channels because I've got chemicals that are opening up these channels and letting in sodium. Other places on the membrane, though, the sarcolemma, I have voltage gated channels. So when we talk about, let's scroll down a little bit, when we talk about voltage gated channels or voltage sensitive channels, these voltage gated channels open up when membrane charge changes, right? That's what voltage gated means. So voltage gated sodium channels are found all over the sarcolemma, and they're found in these things called T tubules. And T tubules, that's probably going to disappear. Let's see. Yeah, it didn't quite fit. Okay, so it's found inside here. Here's the T tubule, which is the folded tube, and here's the sarcolemma. These two places in my muscle fiber, it's generally across its membrane and then down into these folded tubes. These are lined with voltage-gated sodium channels. The goal of these is I change my membrane charge right here. Let's pretend this is the neuromuscular junction. So I spit out some chemicals here. I opened up chemically gated channels right here. Well, I don't need just right here to change its membrane charge. I need everywhere to change it. So I bring in a bunch of positives, which makes this little section of the membrane positive too. Well, if this part starts to go positive, voltage-gated sodium channels will open up and make it more positive. which means that its neighboring section gets a little bit positive and it opens up its voltage gated channels, which makes it positive. So then it makes its neighboring section positive. That's what happens in an action potential. One little place changes its charge, and because membrane charge is what opens the channels in other places along the way, they open up too. So we start to make this part positive and this part positive. We go positive all the way down through across the entire membrane here inside the T tubule. And that T tubule protein, see how we have voltage sensitive tubule protein labeled right here? That's my voltage gated sodium channel right there. It's attached to these little proteins that I see that are here inside the sarcoplasmic reticulum. These ones are attached to the voltage gated channels. When the voltage gated channel opens or it changes its shape, it pushes on these and that causes us to spit out the calcium. So I see all the calcium that's releasing here from inside the sarcoplasmic reticulum. Once I spit out all of that calcium, then my calcium can go down and attach to troponin, which was one of my regulator proteins, right? So I attach to troponin, my push pin, the push pin pops out. When the push pin pops out, the wet spaghetti noodle, that is tropomyosin, it can't stay in place either. It moves away, and now myosin can attach to actin, and we can do muscle contraction. So we started, let's zoom back out. We started up here with releasing a chemical, acetylcholine. When I released that chemical, it changed my membrane charge a little bit. So I used my voltage gated channels to send that membrane charge change everywhere, including these T tubules, which pushed open those mechanically gated channels that allowed me to spit out the calcium I need to make the cross bridge cycle happen.
That's crazy how much happens.
Right? Well, and what's crazy about it too is like it happens without us thinking about it, right? So we take notes, or we scratch our face, or we do something like that, and like we don't even think about this process happening, but it happens in every single muscle cell really fast all the time. So it's pretty cool.
It is. One thing I guess I'm unsure about is how again does it do like the kind of domino effect of along the T tubule and the sarcolemma, I guess, of changing the charge of the voltage charge of each channel. Like,
yeah, let me see if I can find my picture of that, and we can let's pull this picture up here. I know you guys have this one in your notes packet too. Do we know what page this one's on?
Oh yeah, that's I guess it's where is it? Where is it?
I think it's
page 9, I guess.
Page 9 maybe.
Mm-hmm.
Yeah. So this is a picture that you're going to see in the muscles chapter and in the neuron chapter, because both types of cells change their membrane charge using action potentials. I know I mentioned it in the guided lesson, but I'll just reiterate it with you guys since we're here. When you see this word action potential, the second part of its name, potential, literally just means charge. That's what a potential is. It's a charge. This action part of its name, the way I like to remember it is just that it's active, it's moving. So when we talk about an action potential, it's a moving change in charge. It is all an action potential is. So I change my membrane charge somewhere and then I move it everywhere else. That's how muscle contraction works. So what we're looking at right here, so we got these two lines, right? So my first frame up here, this is the sarcolemma of a muscle cell. So over here, we'll say that this is the part of my muscle cell that is the neuromuscular junction. So here, let's draw a little neuron on here. It'll look really funky, bear with me. Here's my little neuron that's coming down. This is where it attaches to the membrane. So this is going to be the place on our previous picture where we had those little green dots, right? That were the acetylcholine. We're going to assume that right here at the very beginning of this muscle fiber, this is where the neuromuscular junction is. So we spit out these little acetylcholine molecules. They attached to chemically gated channels on the sarcolemma of the muscle cell. When they open up those channels, in goes sodium. So sodium rushes inside this cell. Because sodium has a positive charge, it makes the inside of the membrane in here positive. Now, my second frame down here, this is the same muscle cell. So let me draw my same ugly neuron down here. What happened to my plasma membrane though, so here's frame one at time 0. Here's what's happening. I let in sodium because I spit out acetylcholine. Well, wait about 3 milliseconds later, just a very short period of time later, notice that I now have sodium rushing in the neighboring part of the membrane. That's because when we're back up here, when I brought in sodium, this part of the membrane said, oh, check this out. We're positive over here. I'm a voltage-gated channel. I live right here and I'm voltage-gated. It looks like to me, based on the charge of my neighbors, that we're going positive. So I'm voltage-gated and I follow what my neighbors are doing. I'm going to open up my voltage-gated sodium channels and I'm going to let sodium inside. So because this part of the membrane got positive, the neighboring parts have voltage-gated channels and they say, well, my voltage is changing. I'm going to open up. And so they open up and sodium rushes inside. And their neighbors over here that are also voltage gated, they see, oh, we're positive right here. It must mean that I should be positive as well. So these voltage gated channels open up. And so if we had another frame, these ones, the voltage gated channels right here, they would open up next because we've got these positives, the membrane charge is changing, which causes us to change it even more by opening up the voltage gated sodium channels.
Okay, that's helpful. And I guess as much as when I first went over it.
Yes, yeah. So just keep in mind with our types of channels and the way I do an action potential, I start with chemicals, opening up my chemically gated sodium channels. But as soon as they change the membrane charge, everything else that happens is because the membrane charge started to change. So then I'll change this neighboring section, and then I'll change this neighboring section, and we'll do that all through the T-tubule, all throughout the sarcolemma of the muscle cell by using those voltage-gated channels. I think there wasn't there an ordering activity in the lesson? Let me see if am I remembering right that there was?
Yes. Page 11, I think.
Okay, I'm going to pull in my whole lesson outline. We'll see if I can make it big enough. And we'll go to page 11 and see if we can talk through that ordering activity. All right, let's see. Perfect. Okay, so we're here on page 11. So here's a terminology reminder. Excitation, contraction, coupling. We can break it into 2 parts. The second part is the easy part, right? Contraction. That's a muscle contracting for me. So muscle contraction happens. This excitation part means that we've changed the charge on the plasma membrane. So when we talk about excitation, contraction, coupling, we're talking about the steps that it took for me to change the charge on my muscle cell to lead to muscle contraction. That's the big picture. Basically, how does muscle contraction work? Well, it happens by changing the charge and causing my muscle to contract. So when we're looking at all the steps involved in here, what's the first thing that we talked about has to happen to make muscle contraction happen?
Releasing acetylcholine.
Yeah, we got to start with that neurotransmitter, with acetylcholine. So number 1 in this process is my message. My neuron at the neuromuscular junction spits out acetylcholine. So this acetylcholine is an example of a chemical. It's a special kind of chemical that we'll talk more about in lesson number 10. called a neurotransmitter. Wow, that's really big. Neurotransmitter. So it's a special kind of chemical that neurons use to talk to each other or to talk to muscle cells. So my neurons spit out this chemical, acetylcholine. Since it's a chemical, it's going to go down and it's going to open up chemically gated sodium channels. My tip off here is the chemically gated. We're going to open up those channels that open because of the acetylcholine chemical. So I opened up my chemically gated sodium channels. When I open up my chemically gated channels, they make my membrane charge go up. What was that word that we said we used to make our membrane charge go up?
Depolarize.
Yeah, it's that depolarizes word, right? So we depolarize the membrane, number 3. Now, I probably should have, let's add for ourselves. I probably should have put another step or 2 in here. That would have been helpful for us. So I'm going to add a step for us. The step that I'm going to add is what happens after I depolarize the membrane. So once I change that one section of my membrane charge, then it's when we start talking about those channels we were just talking about, right? The voltage-gated sodium channels. Sodium channels, let's say that they move the charge across, I'll have to go down here, across the membrane. They're going to move that charge. I think we should put that in there because that helps me get in my last kind of gated channels, which are those voltage gated ones. So first we saw my chemical acetylcholine. That chemical attached to my chemically gated channels and they opened up. That made my membrane charge start to change. Once my membrane charge starts to change, voltage gated sodium channels take that change and bring it everywhere until we get down to the sarcoplasmic reticulum where they're attached to these mechanically gated channels, the ones that I push on to make them open. So technically, if you're going without this extra step that we added, technically that would be number 4, right? I added the extra step down here, so that's why I'm putting a 5. But we opened up the mechanically gated channels to let calcium out. So I open up those channels, calcium spits out. Once calcium spits out, it's going to go and attach to that pushpin protein. So my troponin protein. And if the pushpin pops out of place, then the spaghetti noodle, that's tropomyosin, moves out of the way. And that's going to allow myosin to attach to actin to make the process of muscle contraction happen. So again, your numbers would be down 1 since we didn't have this step included technically. But to help you make more sense of what we talked about with the action potential, you might want to make that note for yourself that it kind of goes with this step right here where it depolarizes. I have to use these special kind of channels to depolarize the whole membrane.
Okay, I have one question over there. Okay, the number 4 that you just added, it's going to be excitation, exactly?
Yes, yeah, that would be when the excitation stuff is happening. So because it's changing the membrane charge.
Okay, and
yeah, so everything from so 1, 2, 3, and 4, all of those are the excitation steps. Yeah, changing the membrane charge.
Okay, then 5 is I have to consider like contraction.
Yeah, so that, because once we start opening up and start dealing with calcium, the only reason I use the calcium is to make muscle contraction happen. So yeah, that's kind of when that process of contraction starts, is there with step 5.
Okay, cool. Well, thank you for going through that. That's helpful.
Yeah, that's a good way to think about it. I'll put a little note here for us. Excitation equals 1 through 4. Contraction equals 5 through 8. And obviously, if I don't have excitation, if I don't do something right in 1 through 4, then I can't do 5 through 8 either. So that's what we were talking about. Remember when we were talking about Botox, for example, how Botox blocks step 1. If I block step 1, then I can't do any of my other steps. So I can't do any contraction because we messed up excitation at the beginning. Or with sarin gas, instead of blocking step 1, we just made step 1 happen all the time. So when it was time for us to actually stop contracting, we can't stop contracting because I keep receiving this acetylcholine message. So even though the neuron isn't spitting out new acetylcholine, that acetylcholine is still always going down and attaching to these channels. So my muscle cell doesn't know any better. It thinks that we're getting messages, which means we just keep contracting and contracting and contracting, going through all the steps, because we're always doing step 2 when we are exposed to sarin gas.
Okay.
Do you mind to go over the next explanation, please?
So the part down below it.
Yes.
So I'm going to be the mean teacher, and I'm going to ask you guys to help me out to fill it in. Here's what I'll say for you. These things that we have listed here, all of these are actually related to my things that I just put in order. So for example, let's look at our list here. On the list here, we see sodium. So when we talked about the process of muscle contraction, let me go scroll up just a little bit. Which of the steps do we see up here that use sodium?
Second step.
Yep, number 2 uses sodium. So we got those chemically gated sodium channels.
Number 3.
Yeah, number 3 is happening because I opened up those chemically gated sodium channels. And then we got sodium down here, number 4 too, right?
Oh yeah, number 4.
That I added. Sorry, you don't have that one in your notes, right? So sodium is involved in the beginning part where I get my message, but more importantly, maybe the big idea with sodium that we really want to hone in is this word right here, the depolarizing. So we can make a note for ourselves with sodium. The job of sodium is it depolarizes the membrane. Or in other words, remember we said that depolarizes means we're making the membrane charge more positive. I use sodium, the other thing we could put for sodium is I use sodium to make the action potential. That's not all going to fit. Oh, barely. Okay. I use sodium to make the action potential, which is depolarizing the membrane across the entire membrane. That's what an action potential is. So the job of sodium in the process of muscle contraction is it helps me to make that action potential. What could we say would be the function of calcium? What are some ideas about what calcium does in muscle contraction?
The calcium helps to contract, to make the contractions happen.
Okay. Yep. So it's going to help contractions happen. Specifically, what does calcium attach to? Let's remind ourselves about that.
The troponin?
Yeah, so calcium attaches to troponin. Here's troponin over here on the right side, right? Troponin and tropomyosin, both of these proteins, we remember from the lesson we called them regulatory proteins. So when we talk about what tropomyosin and troponin do in the process of muscle contraction, honestly, we could say that these are proteins that block muscle contraction, right? They're my regulator proteins, so they block it. When I talk about calcium, then, you guys told me that calcium attaches to troponin and deactivates troponin. So troponin's job was to make muscle contraction not happen. So if calcium makes troponin not be able to do its job, basically the role of calcium in muscle contraction is to kind of make it happen, right? It's to get rid of the regulatory proteins. So however we want to write that for ourselves, calcium gets rid of regulatory proteins. Those regulatory proteins are these guys over here, right? Tropomyosin, that blocks the myosin sites, and troponin, that holds tropomyosin in place.
Okay.
And when we talk about actin and myosin, these are the proteins that actually do muscle contraction, right? These are the ones that myosin will attach to actin, pull on actin, and actin slides to make contraction happen. So we could say that the function of actin and myosin is just to contract. They're the actual proteins that actually contract. And then there's that one question down here at the bottom, the last one, acetylcholine. What's the job of acetylcholine? Who makes acetylcholine? Let's start there. What kind of cell makes acetylcholine?
Like a neuron or
Yeah, made by neurons.
and it
And
sends,
yep, go ahead.
so it sends the message.
Exactly. Yep. Sends message to contract.
To contract.
This is how a muscle cell knows that it's time to contract. So I need acetylcholine to know that it's time to contract. When I know it's time to contract, I'm going to let some sodium into my muscle cell only for the purpose of changing the charge. Sodium doesn't help me contract directly, but it changes my charge. If I change my charge enough, I'm going to go down to a place where I can push open the channels that help with calcium release. And I need that calcium to get rid of the two proteins that prevented me from contracting. Because if I get rid of those regulatory proteins, now I'm going to be able to do the process of contraction. And that's what actin and myosin do for me.
Yeah.
Did that help, Maria?
Yes, yes, it did. Thank you.
Perfect.
More clear now.
Yeah, thank you so much. That was all very helpful.
Good. Yeah, well, I'm glad that we had a chance to chat about some of this stuff, because this is definitely stuff that it helps to talk through, right? So I'm glad we had some time today.
Okay, thank you.
You're welcome.
Have a nice day. I think
Thank you.
that's all. It's already past 1 o'clock. So.
Yeah, you're good. Happy to help.